Hi folks, this is Dr. Surya Singha, again your lab professor. So now we are moving on to our second lab where we will be introducing you to basics of statistics um, and on how to do sampling, right, and how to sample populations. In this lab, what we are trying to do is um, use different types of beans to represent different type of species and sample them, right? So um, now there are some differences as to how you guys are going to do this lab, particularly since you are doing it virtually. So when you read the lab manual, you will realize that it includes actually collecting samples right in the lab. But in your case, you are actually not collecting data. Instead of collecting data by yourself, we are going to provide you data, right? That is coming from sampling. So now in preparation of this lab, right, I do want you to still read chapter 2. This is the chapter that is titled Introduction to Statistics and Data Analysis. So you have to read it so that you understand how we did the data collection. But rather than collecting data yourself, you will be doing the analysis and doing the worksheet, right, based on the data we are providing to you. So again, you will not do any data collection, but I want you to understand how we collected the data by reading the lab manual. Okay, let's go on to first I'm going to talk to you about very basic principles of statistics, and then I'm going to go over specifically answering the questions in the worksheet and doing statistics related to that particular part. Uh, so um now in statistics or in sampling design, right, I need you to understand several key terms that your textbook or your lab manual also goes through. A variable. A variable is a characteristic or a condition that takes different values, right? It comes through measurements or through counts, right? I mean, think about some examples, right? Um, if we were an actual class, right? The average height or heights of all the students in ecology lab, right? So the heights of ecology students is a variable. Heights of average height of college students in biology, right? Or average body weights of students. Um, all of those are examples for variables. Number of species of amphibians in a particular pond, right? The number of male deer individuals in a given forest. All of those are examples for variables, right? That's pretty easy. Um, Now, population. Statistically speaking, what is a population? It's a particular group, a focal group that you are interested in studying, and by measuring certain variables or counting certain variables, right, is actually called a population, right? Again, a population in terms of statistical sense is the entire whole focal group that you are interested in studying. um, Now let's take an example to make more sense of it. If you're interested in the body weights and heights of all male college students in Massachusetts, right? What is the variable and what is the population, right? Um, well, population is actually in this case all male college students in Massachusetts, right? Um, Variable that you are interested in this case is actually body weight and body height of individual student, male student, right? So variable is something you measure, count. Population is the thing that you are interested in studying, right? Or the entity that you are interested in studying. Now, as you can understand, if population or the focal group that you are interested in studying is extremely large, right? I mean, male college students in Massachusetts. You know how many colleges we have in this tiny little corner of the state. Even that is too big. So generally, at least in ecology or biology, the overall population, statistical populations that we want to study are extremely large, so large that you cannot effectively measure a particular variable. Um, So in order to do this thing, right, if you're going to do this for all college male students, you have to go to every college in the state, find every guy, and measure his height and weight. That's going to take a lot of money. You have to travel all over the state. um you're going to need a lot of help doing this, and that's going to take time. So that's why population level studies are actually logistically challenging. So in order to make the life easy for you, what you do is actually you take a representative sample. A sample by definition, or sample in statistical sense, is a subsample of a population that represents the characteristic of the entire population that you will eventually study, right? So since a sample is smaller than the entire population, that's feasible, that's doable, right? So what you're doing over here is you study a sample, and the inferences you make to the sample, you apply that to the entire population. That's simply how it's going to work. Now so here I'm just like putting all of those together, right? So that's a population, right? That's all individuals, or all individual elements that you are interested in studying, right? And then since it's so big, you take a sample out of it, right, which is representative of the characteristic of the entire population, right? And then so selecting a sample from the population is called sampling, right? And then you actually go and survey that. You collect data from that sample, from that subset, right? And then you do statistical analysis for the sample, but the inference you gather, you apply that, or you generalize that inferences, that statistical outcome, to the entire population. Now let's talk about what type of variables are there, right, that we are used in statistics. There are discrete variables, and then there are continuous variables, right? So discrete variables are actually categorical, right? You can break that particular variable into categories: gender, male and female, right? Color, well, you might argue color has a gradient, but in certain cases you can still assign a particular name to a color, right? Black or white, okay? Like eye color, right? Blue, brown or black, right? So like that, if you can measure a particular variable in terms of discrete items, right, discrete values, then you are looking at a discrete variable, right? So what is the opposite of a discrete variable? Well, that is a continuous variable, right? Examples are anything you cannot assign into distinct categories, right? So this includes length of a particular animal, height of a particular animal, weight of a particular critter, right? So all of those are continuous variables, right? So in a continuous variable, there are infinite divisible, right? A better way to say that is a continuous variable. Continuous variables are infinitely divisible into a large number of units, right? And as a researcher chooses, right, there is you cannot like break it into very distinct categories. Like, I mean, it's quite intuitive, right? um, Another example would be time, right? Time can be measured as a continuous variable as well, right? And if you think about it, continuous variables actually have decimal values, right? 1 hour, hour and 15 minutes, 1 hour and hour and a half, hour and a quarter, right? 1 hour and 5 minutes, like that. Continuous variables have can have a decimal value in it. To make it clearer, we are looking at two examples, right? In the first one, we are looking at, I actually like to keep my pointer in this case, laser pointer. Here we go. So in the first figure on the left, we are looking at continuous data. A scatter plot shows parental mean height against offspring height, right? And both measurements, height, in both cases, those are continuous variables, right? And the best way to represent continuous variables could be actually scatter plots, right? Categorical. Here we are looking at mean distance traveled between male and female individuals of a particular species, right? So male and female, right? The gender, the sex identity is actually categorical, right? You can distinctly assign a particular animal you capture to a male or a female. In certain species, you can argue there's a third category, which is actually the juvenile, right, which have not undergone sexual maturity. Still, those are three distinct categories: males, females, juveniles, right? So if you are plotting categorical variables, you can the most meaningful way is to use a bar plot, right, or a box plot. And now, speaking of statistics, you might have heard a lot about correlational studies, right? Now, what is a correlational study? Or what is a correlation? A correlation test examines relationships between two variables, right? To describe how those two variables behave, right? Now, two variables may correlate, but that does not necessarily mean that two variables there's a causation, right? In other words, variable A and B might nicely correlate with each other, but you cannot say that variable A drives variable B, right? It's maybe it is not, right? So now that's what I meant by causation. In causation, one variable drives the other. Correlation is like, is when one variable increases, other decreases, right? Or when one variable increases, other increases, right? That's correlation. That's a positive correlation, right? And in fact, let me draw you a few things over here and show you what this means, right? Let me actually also get my pencil here. Here we go, right? So over here, right, variable x, variable y, we don't care what they really are, right? So this is actually a positive correlation. When x increases, so does y, right? And then let's take another example over here, right? So in this case, let's do something like this: x, y, same like before. Looks what's happening over here. When x increases, y declines. This is a negative correlation, okay? And let's look at another case over here, right? So when x increases, y remains stable. So that is actually, no correlation, right? X and y are not correlated, right? Another hypothetical situation between x and y. I don't know what's happening with my mouse pointer, right? These dots are my data points, right? So here my data points are all over the figure, right? Again, do you see any clear pattern between x and y variables? No. Again, no correlation, right? So in other words, correlation can be described based on a scatter plot or based on a graph, and it can take all of these four different forms: positive correlation, right? Positive correlation between x and y, negative correlation between x and y. In this one, no correlation, right? Y is stable with increasing x. Same here, no correlation, no discernible pattern, right? Okay, let's move on. So, now I'll give you an example to highlight why correlation necessarily doesn't mean causation, right? Now, before that, if there is actually any dependency between x and y, if there's a causation, right? One of the variables that actually, is responsible for creating the pattern is called predictor variable. It is also known as independent variable, right? So the other variable that actually is driven by, right, that one is called response variable or dependent variable. I think in, like basically you learn these two in terms of independent and dependent variables, right? In ecology, I kind of like to use the term predictor and response variable, right? Predictor is the variable that is driving something, right? Response variable is the one that responds to the predictor variable. Okay, let's take an example to show what is a correlation and why it is not a causation. This is actually real data, right? In the United States, there's a correlation between ice cream sales in summer and shark attacks. All right. Again, in United States, across the entire country, there's a correlation between ice cream sales and shark attacks. You can clearly see it's a positive correlation. When ice cream sales increase, shark attacks also increase. Well, does that mean eating ice cream is bad because you can get eaten up by a shark? No, that's not the reason. Well, the reason is when do you buy more ice creams? During summer. When it is hotter, during the same time, you also tend to go for a swim. During the same time, sharks are more active in shallow water, right? So the obvious correlation between ice cream sales and shark attacks is not driven by a causation, right? Eating ice cream will not increase your chance of getting eaten up by a shark, right? I mean, it's quite obvious. So this is a correlation. There's an underlying driver, that is the summer temperature, right? But it's not causation. It is just a mere correlation. I also want you to understand how to make a good graph, because by end of this lab activity, I will ask you to make a graph, right? So now if you look at this figure, right, there are so many things wrong with it, right? It plots dates and weights, right? First thing, like, I don't know what this is, right? There is no caption. It doesn't describe weight of what and measuring day for what purpose, right? I don't know what is being plotted, right? Is it a particular animal gaining weight, a plant gaining its biomass with progression of time? Well, whatever it is, I don't know. So the number one problem is I don't have a caption. I have no fucking clue what we are looking at here, right? Number two problem is, actually you could also say, if someone was trying to measure time, maybe date is not the best scale over here in the x-axis. Actually, x-axis should have been better labeled as time, which what you are trying to measure, and somebody was measuring it in terms of days, right? It should be like time within parentheses days, right? And the other question is, I don't think day is the best measurement of time if you're only looking between a day and a, if your time scale is only a day and a half. Maybe you should have measured that in minutes or hours, right? So it's a poor choice of the measurement unit and poor labeling of the axes, right? Other problem, weight. Measured in what exactly? Well, weights can be measured in many different things. As scientists, we prefer to measure it in metric scale, right? It's a gram, milligrams, okay, kilograms. Well, and in the US, actually, we use the English terms, English scale units, but we, as scientists, we don't, right? So is it pounds? Well, we don't know, right? So I have no clue what y-axis is actually showing, like what units they are, right? And if you look at the scale, look at 6.45 to 6.95. Well, that's not a huge change, right? So it's kind of, the graphical representation itself is kind of exaggerating, right? The actual change in weights is extremely small, right? Less than 0.5. Well, actually, isn't it exactly 0.5? No, it's actually just 0.4. 0.5, right? The change in weight is 0.5 from beginning of the day until day 1.6, but look how but the weight change is very little, right? So the weight is shown, right? It actually shows as if there is the weight is declining with increasing time, but actual declining of the weight is very subtle. So maybe the actual axis calibration is also a poor choice, right? when in reality you see very little change in terms of weight, the way it is graphed, you will infer, oh, it's a dramatic change, unless you actually read the axis. So there are lots of things wrong with it, right? And also I do see a scatter plot, and I also see a clear pattern, like a negative correlation, but whoever drew this did not actually bother to put a line of best fit to create a line graph, right? By joining the data cloud, right? By creating a point. I'm talking about something like this, like I could actually easily fit a line like that over here, right? Or I could fit a curve like this. The way you fit the line of best fit is a different topic, but I'm not going to go into that detail about fitting a polynomial curve versus a simple linear regression. I'm not going to talk about that, but I'm pointing out the fact that it doesn't have a line of best fit. So there are many problems wrong with this graph, and I want you to take a look at this graph and think about it, right? And try to figure out, how do you make a complete graph, right? So moving on to other definitions of the statistics. Parameter, right? Parameter in statistics is a numerical description of the population you are studying, right? So the most widely used parameters in any scientific discipline for that matter, and most definitely for biology, is a average, two standard deviation, right? Well, there are other similar measurements like standard error, right? Spread, median, mode, those are also parameters. Now, you know what average is? It's a middle value, the kind of the intermediate value, right, in a given data set. And standard deviation, you also know that. Standard deviation tells you like how far each measurement is from the mean. That's what standard deviation is, right? So since standard deviation tells you the deviation or the shift of a particular data point from the average, it also tells you, like, for a particular random data point, like how different that data point is from other rest of observations, right? So that's why standard deviation is important, because it actually tells you something about the spread of the data. All right. Another parameter is variance. And variance and standard deviation are very interchangeable terms. They don't mean the same thing, but they're very related, right? So standard deviation is the square root of variance, right? So both of them effectively capture the spread of the data, right? How far each data point is spreads around the mean. That's what it tells you, right? Very simple stuff. So, well, another term, statistics, right? There are two kinds of statistics. One is descriptive statistics. Now, a parameter characterizes the population. A statistic characterizes your sample, right? So, statistics and parameters are interchangeable terms. So, mean, standard deviation, variance, mode, all of those, standard error, all of those are also they are not only parameters, they are also statistics, right? And you might have realized that we are using different notations. It's simply consensus that we use these notations when we are talking about statistics versus parameter. So I don't want you to know about these notations, but don't get scared by it. Don't like try to wrap your head around why there are different notations. It's not important. And test statistics. Well, now so descriptive statistics is actually what I just mentioned, right? Mean, standard deviation, variance, all of those are descriptive statistics that describes the characteristics of your sample. Test statistics are actually statistical numbers, numeric values you extract after running a particular statistical test on the entire set of data, right? And I'm not going to go into detail about describing what that is, because we are going to do one of these, right? So for example, you might have heard about hypothesis testing. You collect data And then based on the data you collect, you produce a hypothesis, or in the other way around, you actually are interested in a particular research question. So based on that research theme, you develop a construct a hypothesis based on existing knowledge. Then you collect data and you test the hypothesis using statistical tests, right? T-tests, F-tests, chi-square tests. Those are some examples for statistical tests. ANOVA is another statistical test that you may have heard of, but I'm not surprised if any of you have not done this before. But that's fine. That's why we are here here in this class. And then you apply these particular statistical tests to validate the hypothesis, and then when you at the end of the statistical test, the test spits out a particular number, right? That number or that numbers are referred to as test statistics, which helps you to determine whether your hypothesis is valid or not. Now again, we will go through test statistics with respect to an actual example, right? In another statistically important terminology that I want you to know is confidence intervals. Now normal distribution, that's something you should be familiar with by now, because this is something you always you might have talked about during high school and all that. So statistically normal distribution, when you plot it, right, the frequency versus a particular the values you are measuring, right, the graph looks like a hump-shaped curve, a well-balanced bell-shaped or a hump-shaped curve, right? And then you can, and then what you do is, for that particular variable you measured, right? So on this axis is a variable you measured, right? It could be like a length measurement or a weight measurement. On this axis, the axis that I haven't drawn, is actually the frequency, right? That a particular measurement is made, right? And this notation mu stands for mean, right? Or average, right? So this mu indicates this is actually the actual mean of that particular sample, right? So it's a statistic, right? And what you do is actually, with the mean, you can also measure standard deviation for that particular population, right? And what we are doing over here is calculating confidence intervals around that particular mean, right? So if you add or subtract, right, a single standard deviation from and to the mean, right, you actually are looking at 68% confidence interval around the mean, right? And then, so it's actually this green component, right? That is 68% confidence interval. What does that mean? That is actually a single standard deviation around the particular mean, right? So this is actually one standard deviation, right, around the mean. This is added to mean. This is one standard deviation subtracted to mean. So this particular range of data corresponds to 68% confidence interval for that particular data set. If you add and subtract two standard deviations, right, add and subtract as two standard deviations around the mean, you are looking at 95% confidence interval, right? That is actually this entire data range, which includes green and this purple. Okay, now 95% confidence interval is kind of the standard we use for most of the statistical tests in sciences, most definitely in biology, and most certainly in ecology. Okay, if you add and subtract three standard deviations, right, from and to the mean, you are looking at 99.7% confidence intervals around the mean, right? So that actually includes, this entire data range, which includes the green part, the purple part, and this gray part, right? So what is the importance of calculating these confidence intervals? Well, that actually helps you to understand whether how much of your data points are captured by these standard deviations, captured by these confidence intervals, right? If you collect 100 data points, how many will be captured within 95% confidence interval? How many within a 68%? How many within 99.7%, right? So it's kind of, that's what you're looking at over here, right? So it gives you an idea about the anomaly of your data points, right? So any data point that is actually within outside, if any data point that is located outside 99.7% of the confident intervals are actually extremely abnormal data points, right? Those data points are actually outliers, right? They're probably an unreliable measurement, okay? Could be a measurement error that occurred when you actually collect the data, right? And so 95% confident interval, data points that lie within the 95% confident interval, that is two standard deviations added to or subtracted from the mean, right? That's a reasonable range, right? You will expect a lot of data points to fall within that particular area, that particular range, right? So and that usually captures most of your observations, right? And any observation outside the 95% confident interval, well, those again could be due to some measurement error or some anomaly, some abnormality in that particular measurement made, right? Now remember that this kind of confident intervals can only be applied to data sets that have a normal distribution. If you have a data set with a non-normal skewed distribution, like this, let's say, right? You can't apply the confident interval, right, theory. Or if anything looks like something like this, you can't apply confident intervals like this, right? Right. So any of those red graphs I draw, those are non-normal distributions, those are highly skewed distributions, right? And we will not apply confident intervals to those, right? You can mathematically, but you cannot collect statistically robust inferences, conclusions based on by calculating confident intervals for non-normal data, right? So again, 68% of the data, 68% confident interval refers to data that is located within a single standard deviation around the mean. Again, 95% confident interval refers to data points that is found within two standard deviation around the mean, added or subtracted. Last, 99.7% confident interval refers to data points that are located within three standard deviations around the mean. Okay. So confident intervals tells you, like I kept repeatedly mentioning, gives you an idea about the dispersion of the data points around the mean, right? So, and like I mentioned earlier, 95% confident interval is the universally accepted confident interval. Okay. And so in future, I'm not going to ask you to calculate confident intervals for 99.7%, 98.5%, about 68%. No, I'm going to, when I say calculate confident intervals, I'm going to assume that you know it is 95%, okay? So 95% confident intervals, let me give you an example about this, right? And kind of it will make sense to you, right? Let's say that you're actually measuring body lengths of a particular crab species, blue crabs, right? You go to Florida, you deploy traps and you capture blue crabs. Is a really cool species, and then you measure their carapace lengths, right? And after you measure all of those, right, let's say you caught 100 individuals and then you measured all of their carapace lengths, and after measuring all the carapace or body lengths, you calculate the mean, and then you calculate the standard deviation based on mean and the standard deviation. You calculate the 95% confident interval, which happens to be 100. That is the lower confident interval. That's mean minus 2 standard deviations. And 200 is the upper confident interval. That is the mean plus 2 standard deviations, right? Oh, by the way, these are like just numbers I pulled out of air, right? Those are not real numbers. Now, how do you actually express that in a statistically accurate way, right? So I'm actually going to give you like two options over here, right? What would you think the best or the most the ideal way to express that? It's actually B, right? You might have heard through other classes or maybe through high school that it's number A, right? There is a 95% probability that the population mean of blue crabs fall within 100 and 200 millimeters. I'm not saying that is wrong, but it's not I have a hard time as a ecologist wrapping my head around that statement, right? But what it actually tells you is this, right? Think about what we are doing over here, right? You are interested in not just a small sample of blue crabs. You're actually interested in the entire blue crab population in Florida, in southern Florida, right? So what you are trying to do is get an idea about the body size of all blue crabs in southern Florida beach, right? But you cannot do that for all crab in every freaking crab in southern Florida. So you do a sample, right? So the correct way to interpret that is if you collected 100 samples, right, of blue crabs, right, and you measure the body lengths of all of those 100 crabs, right, and 95% of those crabs you capture, right, or 95 crabs you capture, right, will fall within 100 and 200, right? That's what actually that confidence interval mean. Okay, let's look at that in another way. Remember, like you collect data for a sample, but you're trying to reach conclusions about the population, right? So if you collect 100 crabs, right, measure lengths for all the 100 of those crabs and calculate the confidence interval for all of those, right, at least 95 of those measurements you took, right, will capture the actual population mean, right? Remember what population mean is. Population mean in this case is the body size of average body size of crabs in southern Florida beach, right? So there you go. That's what actually confidence interval means, right? All right, let's move on. Right, now in this example, let's look at two different states in the country, Washington and Florida, and we will measure student IQ in those two states, right? So when you are looking at any biologically, scientifically interested question, you need to come up with a hypothesis, right? So biologically or scientifically, you may expect to have a different IQ differences between Florida and Washington, right? Maybe Washington schools have better education, right, and they have more money, right? So maybe because of that you will expect Washington students to outperform Florida students. So the null hypothesis should actually take an opposite turn than what you expect, right? If you expect a significant difference between those two, you say null hypothesis says you would not expect a difference, right? In this situation, based on economic situations, based on the commitment of teachers, based on the commitment of the state governments, you would expect Washington students to do better than Florida. So your null hypothesis says the average IQ in Washington will not be significantly greater than the average IQ in Florida. You can also state something very similar, like the average IQs in Washington versus Florida will not be significantly different. Now what you do is you actually go to those states, you go to several schools and collect data, and then you calculate the average, right? So Washington IQ average, let's say it was about 90. Okay, in Florida it turned out to be about 80, where you figured that Washington IQ is about 10 points higher on average than Florida. And then any statistical application also spits out something called a p-value or a probability value. And in this case, you got a probability value of 0.34. You know, if it is harder for you to wrap your head around a probability value, make it a percentage. At about 35%, right? About roughly about a third. Now, what does that probability value means, right? What kind of a probability is this? Probability of what from happening? Well, think about what I mentioned about the null hypothesis. Think why we have a null hypothesis. Because when you do a statistical test, when you calculate test statistics, when you calculate probability for that particular test statistics, you are actually testing whether that null hypothesis is true or unacceptable, right? So given that the null hypothesis, which is the average Washington IQ, is no greater than that of Florida, if that is true, then there is a about 35% chance that we would measure the IQs at least 10 points greater in Washington than in Florida, right? Well, what does that mean? Let me break it down again, right? Even if the null hypothesis is true, right? Meaning what? Even if truly Washington I-caves are not significantly great, even if Washington I-caves and Florida I-caves are not that different from each other, right? If you were to actually measure the IQ in those two states, right, there is nearly a 35% chance that you will end up actually getting a higher value for Washington than in Florida, right? So that 20 not 20 point, I cannot do math, the 10-point difference, 90 minus 80 is actually 10. So that 10-point difference that you actually calculated in your survey, right, that 10-point difference is not statistically significant if given this particular p-value, right? Another way to interpret this is actually that although there is no significant difference between Washington and Florida I-caves, right, by chance, by random dumb luck, you might end up, right, getting a greater value for Florida, greater IQ value on average for Washington than for Florida, right? So regardless of the gap that you actually end up with, right, that particular difference is not significant, right? So the 10-point difference is actually insignificant. It's negligible based on the statistical analysis. So p-value, like another way to look at that is actually the probability of observing a difference even if no statistically meaningful difference exists, right? So if IQ in Washington, right, if IQ values in Washington are not actually higher, right, we would still measure at least a 10-point difference between Washington and Florida, at least 35% of the time. 35%, if you convert that into a fraction, it's about a third, right? And that kind of probability, like we call it, like it's because of the random noise, right? Kind of we kind of by chance, right? That 10-point difference that we observed maybe just by chance, right? It's not realistic. So now lower the p-value that your statistical program spits out, right? It is less likely, the differences you observe is less likely to be by chance, right? And higher the p-value, right? Higher the p-value spits out by your statistical program, right? The higher the likelihood that the differences you observe are by chance, right? Now this, how do you determine what is the breaking point? What is the threshold to determine the differences are significant or not? Like what p-value, right, do you use as this cutoff mark, right? So that particular cutoff mark that you use for p-value is also known as alpha, right? Now alpha, right, is actually also related to the confidence interval we earlier talked about. You might remember that I mentioned, like when you calculate confidence intervals, at least in biological sciences, the universally accepted confidence interval is 95%, right? So alpha is actually 100 minus your confidence interval, right? 100 minus 95% is what? 5%, and 5% as a decimal value is 0.05. So your cutoff point is actually 0.05. If the p-value that you calculate through a statistical program is lower than 0.05, then the differences you're observing, or the values you're comparing, are statistically different. Okay? If the p-values that your statistical program kicks out is greater than 0.05, then there are no statistical, there is no tangible evidence to suggest that the numbers you're comparing are different, right? So again, let me go back, right? Your accredited or accepted alpha or p-value, or the cutoff p-value is 0.05, right? If the probability value that you calculated through a statistical analysis is lower than 0.05, right? If the statistical value you calculate is below 0.05, then, right, the two things that you are comparing, the two averages you are comparing, the two entities you are comparing, right, are statistically different, right? Significantly different. If the p-value you calculated is greater than 0.05, greater than 0.05, then there are no statistically significant difference between the two things you are comparing, right? Now let's try to understand the same principle of statistical analysis with respect to more examples, right? In this example, right, I'm trying to tell you, why do we need statistical testing? Why cannot we just compare the averages, right? In the previous case, note that Washington, in our hypothetical example, came up with a greater IQ average than Florida, right? But the statistical test revealed that particular difference as insignificant. So you need to understand that just comparing averages is not enough. That is exactly why we need to go towards additional statistical tests to see if these two samples are actually different, right? In this example, you are looking at a population of first graders, okay? And you have two samples of the first grader, sample A taught by a particular teaching method A and sample B taught by a particular teaching method B. You now have your sample sizes are not exactly the same, they are slightly different, okay, but comparably good, right? And but comparable or very similar. And then what you do is, after exposing the two samples to two different teaching methods, you give them a test, give them an exam, and then you calculate their average. Sample A taught with method A averaged at 76%, okay, and the sample B that was taught with method B got 71% as an average score. Now I know that 76, right, is greater than 71. Yeah, so can I actually conclude reasonably without any doubt that teaching method A is better than teaching method B? No, because look at what I'm plotting over here, right? Let me take my pointer. There we go. Look at these graphs, okay? Although sample A has an average of 76, sample B average of 71, right? The spread of the data, right, is actually like more or less similar, right? They both actually, in sample A, the actual scores of these kids of these first graders range between somewhere between 70 to 80, right? Slightly over 70, but slightly over 70 and slightly over 80, right? And in the sample B, it's actually somewhere between 65 and 75. Well, that's actually, those ranges are not that different from each other. 70 to 80 versus 65 to 75, that's not a whole lot of different. So although average is greater, the ranges are quite comparable, right? So that's why actually, since the data spread matters when you're making a statistical inference, the average itself is not good enough, right? You need to look at the overall spread of the data. That's why we go for these, statistical tests and calculate the probability of the statistical probability of one truly being greater than, or one truly being significantly different from the other. So now, what kind of inferences, by looking at the spread of the data and the averages, what are the inferences you can gather, right? So the sample data show, right, a 5-point difference between the sample A and sample B, between the method A and method B, right? However, right, there are two different ways to interpret this, right? There is actually no significant difference between the two teaching methods, right? And the differences you observe, the 5-point difference, may be actually meaningless. It's the difference in itself is insignificant because the spread of the data, the actual score ranges, are not that different from each other, right? That's one conclusion. The second conclusion is actually, well, that 5-point difference may be actually meaningfully different, right? That's 5-point difference is good enough to be significant, right? We don't know which way to go, right? Inference 1 or 2, right? In order to be certain whether it's 1 or 2, we need to conduct statistical tests, right? So in other words, right, we need to avoid sampling errors biasing our inferences. Now this is a good way to think about what sampling error is. What is actually sampling error, right? So if you read over here, it says there actually is no difference between the two teaching methods, and the sampling difference is due to by chance, or the score difference, the 5-point difference is simply dumb luck. That's what it means, right? And we say that the difference is actually not driven by the teaching method, it's simply the sampling error. Now my question is, what the heck is sampling error? Sampling error is actually this, right? So if it is not the teaching method, what other possible reasons could have resulted the differences in average scores, right? Now that's what sampling error is. So what do you guys think? Well, someone can argue that, well, sample A and sample B are not equal, right? Sample B is 1 less than sample A. That unequal sample size might have created an error. Well, okay, that may be, that could be. I'm not saying that is, but it could be. That's a source of sample error. Well, I did not mention that sample A and sample B had this similar. I did not say that earlier when I described this example. What if kids in sample A are actually smarter than sample B? What if that is the case? That's a sampling error, right? So when you assign sample A and sample B, when you pick sample A and sample B, if you didn't randomly pick kids for A and B, right, you might end up having, smarter kids in A or B. In this case, what if the average score is because those kids were smarter? Right? So that's a sampling error, right? Not if your comparative samples were not coming from, if the sampling was not done randomly, right, you will end up in a sampling error. Well, another source. What if the teacher that taught these two methods are not the same, right? If you use two different teachers, one is a great teacher, other one is a bad teacher, then of course, maybe the sample A got a great teacher. Maybe that's why they have a higher score, right? And sample B may have gotten a bad teacher, okay? And I also didn't mention that we gave the same test to A and B, right? If I did not give exactly the same test, right, that could also create a sampling error, or better yet, it's a designing error, right? And most importantly, sampling error comes from actually your sample not being representative of the entire population, right? If the actual population of the first graders were about, let's say, 1,000 kids, right? And if our sample size is less than is 22 or 21, well, that's actually a really small sample, right? A sample of 21 is not comparable to a population of 1,000. Right? So most likely this sample is way too small to represent the entire population, right? That could create a sampling error, right? Remember that each kid is different, right? So unless your sample is not great enough, you will not experiment a good proportion of the population, right? So if your sample size is too small compared to the population, right, you will end up with random errors, right? And which also results in sampling errors, right? Now that is actually one of the biggest sources of sampling error. Again, let me repeat. Guys, when I repeat it because it's extremely important for you to understand, right? That's why I will keep saying same thing over and over and over, okay? If your sample is too small to be representative of the population, you will end up in random errors in your test, in your design, in your research, and that will lead to sampling error, right? So it is all of those reasons I just explained to you that you need to go for statistical testing to see for significant differences. That's why just comparing averages is not enough. That's why just eyeballing the range of the data is not enough. Now, let's try to understand sampling error furthermore, right? The reason is it is really important to understand. Now, sampling error, right, is not the same as human error. That's not the same, right? Let's say, could incorrect grading be a source of sampling error? No, right? If the teacher fumbled, made a mistake in grading, that's not sampling error, right? If somebody made an error in doing the statistics, that's not sampling error. Human bias is not sampling error, okay? Again, sampling error comes from your sample not being representative of the entire population. Your sample being too small to represent the entire population, right? Your sample being a biased subset of the population. Your sample being not a random subset of the population, okay? Now let's try to understand this a little further. Right, what are we doing here? So you take two samples from a particular population of 1,000 college students, right? And the average and the parameters of that population, the average age is about 21 years, slightly over 21 years old, right? College kids. And the average IQ of those college kids is about 112.5, and that population contains about 65% girls and about 35% males, right? You already have a female biased college population, which is not surprising actually. And then let's take a look at the samples we collected over here. Sample 1 and sample 2. Sample 1, Eric, Jessica, Laura, Karen, Brian. Sample 2, Tom, Kristen, Sarah, Andrew, John, right? So sample 1, average age is about 19.8, 20 years old, right? And in sample 2, the average age is about 20 years old, right? And well, I know that the two ages are not perfect, right? Well, both are, slightly below the average, but they are not that different from each other, right? So the sample averages are actually comparable to the population average, okay? So that's actually good. How about the average IQ? Average IQ of the college kids are about 112. Sample 1 is way below. It's about 104. Sample 2 is actually slightly above average, right? So the average, although the sample 1 and 2 are comparable or equal in terms of the average age, they are not comparable in terms of their IQ. Now let's look at the sex ratio, 65 to 35 in the population, right? In sample 1, 60-40, right? In sample 2, it's completely the opposite, right? Sample 1 has more girls and sample 2 has more boys, but the population has more girls, right? So sample 1 is actually a better representation of the sex ratio of the population, but sample 2 is a completely fucked up sample compared to the college population. College population, more girls. Sample 2, more boys. Yeah, that's a really bad sample, right? So that will lead you to commit sampling errors, right? And average IQ, right? Sample 2, yeah, that's actually comparable to the college population. Sample 1 way below the college population average. So there are two reasons to throw away both sample 1 and sample 2. Sample 1, I don't like it because its IQ is fucked up, right? IQ is way below the college population average. Sample 2, I don't like it simply because its sex ratio is not comparable to that of the population, okay? So generally, right, when you take samples from the population, you will have some differences, right? You will never have a sample that is identical to the population, right? So the question is to minimize these differences between the population and the sample. Again, your sample will never be identical to the population. They will be always different, right? But you have to try harder to make sure that your sample closely resembles the population in terms of parameters, in terms of these features that we are interested in, right? So in other words, the discrepancy or the differences between the population's parameter and the sample's statistics should be minimal. Okay? So the question is, how do you decrease sample error? How do you make sure the discrepancy between statistics and parameters are subtle, right? Well, survey multiple samples, right? Collect samples over and over and over, right? Another way is actually increase your sample size, right? Right. In this case, 100,000 college kids, but your sample size is what? 5 kids? That's stupid, right? That is all not going to work. Right? If you have 1,000 college kid, you probably want to actually represent your sample 10% of the college population, right? Like 100, right? If it is 50%, it's great. If it is 20%, well, that's also acceptable, right? So, higher the sample size, the better it is, right? The better it helps to minimize sampling error, right? Greater the number of the times you sample, the lower the sampling error. Okay, so that's how actually you minimize sampling error. That's how you minimize the discrepancy. Okay. Let me explain another reason why you shouldn't rely on average alone, right? When you're comparing different samples. Look at these samples represented by the red, green, and the blue lines. All right. They have exactly the same mean. Look at their mean values over here. Let me take my pointer. Right. They have exactly the same mean, a mean of 0, right? Their mean perfectly overlaps with each other, right? But do they have the same standard deviation? Do they have the same spread of data? The red sample has a very narrow distribution, very small standard deviation. Green one slightly greater. The blue one much wider spread, meaning much wider standard deviation, much wider variance, right? Well, if you compare the red or the green or the blue with the pink data set, yeah, they have actually not only different ranges, not only different sample, different standard deviations and data spreads, they also have different means. So that in that case, yeah, it's reasonable to it might be reasonable to look at the averages, to compare averages. But even between the pink and the red, you do see a significant proportion of overlap between those two data sets, right? And if you compare the green and the pink one or the purple, whatever that color is, right, you see some overlap, right? If you compare the blue and the purple one, there's a much overlap between those two data sets, right? So the point is, the average itself does not tell you much about, does not help you to come to a statistically robust, rigorous conclusion about the data, right? Some data sets might actually have, right? I mean, this is really interesting, right? Blue, green and red, exactly the same mean, but completely different spreads of data, right? So by looking at the mean, you cannot say, oh, those samples are the same, or they are comparable, they are similar, they are significantly not different from each other. You cannot say that. The spread is completely different, right? Then you look at this purple or pink data set against the blue data set, right? They have completely two different means, right? They're not even close to each other, but their data spread overlaps completely with each other, right? The blue data set completely overlaps with the entire range of purple data set, right? So actually, those two may not be statistically different from each other because of that overlap, although their means are different, right? So having same mean or different means does not necessarily tell you that your samples are significantly different from each other. That's the point I want you to learn. Well, that's actually the conclusion of the statistical introduction. Now let's navigate on to actually doing the lab and get the fun stuff over with.